In terms of human rejuvenation, although most people are focusing on epigenetic reprogramming, there may be a better approach and one that is closer to clinical use. With that in mind, today's guest is Dr. Colwyn Headley, who has used mitochondrial transplantation to improve cellular function. So welcome, Colwyn, and let's get into the presentation.
I'm excited for this. So I call my presentations yap sessions because it's really just be yapping most of the time. It takes a village to get other stuff done. I like to just get this out of the way because I always forget sometimes. And this is probably one of my most up-to-date acknowledgement pages. This is stuff I started at Ohio State and sort of transitioned from Texas to now Stanford. Mitochondrial dysfunction to me, I view it as engine failure. Check engine light comes on. You take your car to a mechanic and you can put a new engine in that vehicle, keep using it for another couple, thousand, maybe 10,000 miles. And I think in principle, this is something that we can do for the body. But before I start talking about the engine, I think, or talking about transplantation, I like to just quickly touch on how the engine breaks, or some of the modalities that influence the mitochondrial dysfunction. The mitochondria, the electron transport chain contributes to ATP production. As the electrons kind of go through this roller coaster, some fall off, unfortunately, or they're necessary. So from complexes 1 and 3, you have some leak of electrons, and because you've got oxygen sort of everywhere, these react and you get a superoxide, but we're just going to say reactive oxygen species for simplicity. I consider Ross to be kind of, it's like a drunk guy at a bar, maybe too much alcohol, and he's ready to fight. And they're super reactive. They like to attack anything that they can, which is proteins, lipids, DNA, RNA, whatever they can attack to. And within the matrix, you've got copies of mitochondrial DNA. You've got a couple hundred to a couple thousand, depending on the cell type. And under typical conditions, you also have the bouncers, which are like the antioxidants. You've got catalase, superoxide dismutase, glutathione peroxidase that are really helping in nullifying or in keeping a proper ratio of ROS to cell activity. And you also have these repair enzymes, DNA polymerase complexes, that when ROS levels are normal or within a proper threshold, you don't see a lot of damage being introduced by DNA polymerase. But when ROS it becomes more like a mosh pit. There's so much more oxidative stress. Everything gets hit. Everything is getting like knocked out. The enzymes are supposed to help with detoxifying the mitochondrial DNA. The stuff that's supposed to repair it also increases its sort of like error rate. And you have accumulation of mutated mitochondrial DNA. And this is just some of the flavors that contribute to mitochondrial dysfunction from, I guess, the purview of mitochondrial DNA mutation accumulation. This is the perpetual ROS sort of death cycle that I like to use because it's really interconnected. The amount of, if there's an increase in ROS within mitochondria, typically there's a pathogenic shift towards more mutated mitochondrial DNA, which then ultimately influences the electron transport chain. Or another way to, I think people call this like heteroplasmy, and pathogenic heteroplasmy really, where it's this like continual shift of, as you get older, if we consider your cells to be cars, the engine is always running. Like you never turn that stuff off. So over time, that's just the mat starts to mat, and you can't outrun a bad engine. And so another way I think about it is in terms of the Pac-Man. You can think of ROS as the little balls, and then the antioxidants are like the Pac-Man. And because mitochondria is probably one of the major contributors of oxidative stress inside the cell, if there's increased mitochondrial dysfunction, there's going to be increased oxidative stress. And aging, to me, or how I think about it, is a young versus an old cell is really just this misbalance of ROS being produced versus eliminated, and then sort of chronic conditions pushes you over the edge. And if it becomes too if this sort of bias becomes too great, it's like game over. That's it. And so mitochondrial transplantation, again, this idea that you can kind of just, the same way that you can go to a mechanic, swap an engine in, and keep using that vehicle. I think it's been an area of growing interest where one has been organically shown by, well, I'll get into this. So there's a couple methods by which this transplantation stuff has been kind of characterized. Like there's the tunneling nanotubes where cells will make these kind of protrusions and they'll siphon, or it seems to be going both ways, but they'll trade mitochondria. There is also encapsulation, either in the form of mitochondria in microvesicles, or we have the ability to make lipid nanoparticles in the liver as well. And then we also noticed that mitochondria are they have this, I call it kind of like a secret handshake, because we don't know all of the nitty-gritty mechanistic, like, things that happen for internalization, but they seem to be taken up by the environment. And then depending on the cell, it's like a different handshake. So there's, like I said, a growing interest in this space because of just the universality of mitochondria. There's almost every cell except probably mature red blood cells use mitochondria either for energy, for its energy portfolio, or for some of the signaling modalities. So when I was in grad school, I started grad school in 2015. Damn. Uh, when I was in grad school, one of my projects was looking at the impact of aging on T cells, really trying to understand. Well, it's kind of already been characterized that with aging, there's a decrease in mitochondrial function, there's an increase in oxidative stress. And this sort of correlates with a decrease in ability of HT cells. If they're responding to the same sort of cognitive antigen, those HT cells don't express the same markers of activation. They don't produce the same magnitude of cytokines, and it has a bias towards inflammatory cytokines, and they don't proliferate to the same capacity as a younger T cell responding to the same cognitive antigen. And I like simple stuff. We got to this idea where it was like, why don't we just replace the mitochondria instead of trying to dose with antioxidants or different, or trying to modulate one or two pathways within the mitophagy or biogenesis. If we can replace it, then we'd hope the cell is smart enough to figure out how to get rid of the bad stuff. And so, in we the method that we came up with is we figured out that centrifugation is simple enough to do it. You can isolate your mitochondria, mix them in culture with whatever cell type, put them in a centrifuge for 5 minutes, and that's it. Like, to me, that was gold mine, because I like simplicity and I like reproducibility. I never want to be in a situation where someone's like, "Oh man, this protocol don't make any sense." You literally just need a centrifuge. Like, centrifuge, that's the most expensive part, probably. Like, so, and it's easy to scale because we can just change the ratio of mitochondria to cells within suspension. And so we're able to scale it to, almost 100% for transplantation. And so we could really do experiments using this sort of framework. And so it was like this: you culture cells, isolate their mitochondria, and then centrifuge them with your immune cells. And so we narrowed down on CD4 T cells, particularly because my PhD was in immunology, and we're looking at sort of the uh, consequences of adaptive immune aging, particularly with T cells in context of tuberculosis, one of the major bugs that I studied. Again, this sort of like the Ross debt loop that I mentioned. And so after sort of optimizing the protocol, we just went to work. We would take mitochondria from fibroblasts. For the mice studies, they were coming from mouse embryonic fibroblasts. We essentially made a sort of a calibration curve to standardize how much mitochondria we'd isolate from a specific amount of fibroblasts, and then just standardize that for all of our studies. And so what you're seeing here is the young CD4 T cells, and then versus the old, and then the old after mitotransfer, mitochondrial transplantation. And we saw a significant increase in oxygen consumption. And again, I like the seahorse because it really does, it's a bona fide way to measure oxygen tension within like a culture. But because oxygen consumption and oxygen utilization, like it's OXPHOS activity is sort of coupled to ROS production. So if you measure an increase in oxygen consumption, you kind of have to also make sure that this is not related to a significant increase in reactive oxygen species production. So I learned this technique in undergrad called electron paramagnetic resonance. It's probably the most sensitive way to detect free radicals, because kind of like trap them, and gives you like an extended time to characterize them, and then based off of the intensity of the signal of the spin trap, we can sort of infer how much based off the intensity, but and also based off of the probe that we're using, we can infer how much superoxide is being produced by mitochondria in T cells. And so this is a comparison of a young CD4 T cells. These are the old and the old plus mitochondria. So we saw an increase in oxygen consumption, but a concurrent decrease in superoxide production. We also did some proteomics and looked at some of the the most upregulated gene ontology pathways related to mitochondria and the cell itself. But what we noticed was things related to the architecture. So the mycose complex and cristae formation were some of the top hits. And so we did electron microscopy to take a look at the mitochondria look at the mitochondria inside T cells that didn't get any transplantation, and then these are after transplantation. And I kind of, the difference is like going to the gym and you can see your abs. Like that's look at that. It's just it was amazing the first time I saw it. And again, because it's such a simple thing, like we did, like every time I present, I tell people that it's not rocket science for the fact of how simple this was to just do. And but the magnitude of impact that it had is exciting. And so we were able to show that you could improve OXPHOS, reduce oxidative stress, and we went on to sort of compare the functionality of a sort of the old T cell or the old T cell after it's sort of been biogenically reprogrammed after transplantation. Would that change activation, microexpression? Would that have an impact on cytokine production, and would that lead to improved proliferation? And so again, because the mitochondria is such a it's an engine, but it's also a CPU. Nothing really happens without its permission. I already anticipated that because we saw these sort of changes in redox activity, we would see changes in activation markers and stuff like that. And that's exactly what we saw. We saw increase expression of CD69, CD20, co-expression of CD69, CD25 on CD4 T cells after transplantation. So these are paired experiments, meaning that the cells came from the same animal, and then they were either just given mitochondria or not. We looked at cytokine production, particularly IL-2, which is known to go down with aging, and we saw an increase in that. And then we also looked at proliferation. And so to me, again, this is all ex vivo, but if you can augment the activity of a cell that easy, then the obvious step is to go in vivo. And so one of the experiments that we did, two experiments we did a lot of experiments, but I'll just try to be brief. We wanted to show that, at least I wanted to show that this is like a disease agnostic sort of stuff. If you fix the T cells, it doesn't matter what the condition is, it's going to have a benefit. And so we chose two models, a viral infection, influenza, which old folks get a lot too, and then tuberculosis, which tuberculosis is a aerobic what's the word? It's a bacterium that it's in the air and you get infected that way. But typically, most competent people will not develop a hardcore infection until later in life. The bacteria goes latent, and then when you're older, because of your immune system being all wonky and your engine crapping out, chronic inflammation tends to reactivate the bacteria, and so it's another aging associated pathology. Not big in America, but I'm from the Caribbean, so so when we looked at influenza, well, maybe I'll give a little bit more context. But so for the influenza, we particularly looked at mortality. Look, we either gave them younger mitochondria or didn't, and then after infection with influenza, we just tracked primarily mortality. But then with tuberculosis, we looked at the CO2 burden in some of the organs that we expect to see TB, so lung, spleen, I think, and liver. For influenza, we saw that there was a significant increase in the survival of the Ragnac mice that got the old CD4 T cells after transplantation versus old CD4 T cells with nothing, or then Ragnac mice didn't get any CD4 T cells at all. And then for tuberculosis, we saw a better control of mycobacterial burden in lung and spleen in the Ragnac mice that got the old plus mito combo. In tuberculosis, and I mean other conditions as well, there's exhaustion. So exhaustion happens when sort of like your immune system is constantly fighting, and it just can't deal with the it's getting beat up. Like you are not like you're trying to get up and it's like getting punched. Mike Tyson. That's really what I think about. And beyond exhaustion, there's this sort of senescence. How I kind of look at it is, or another way that I think about it is like exhaustion is like your car dies on the highway. Senescence is like you never actually able to leave the parking lot. Like you, either way, you're gonna have a bad day. You're gonna have to call for some help. And so with tuberculosis, exhaustion is one of those things that the longer you're infected, the more you have a profile of exhausted immune response. And some of those markers are PD-1, TIM3, like 3. And from some of the data that we had, we just screened for change in exhaustion-associated proteins. And so we compared the old versus the old plus mito, and we saw there was this shift. And to me, that was okay. Let's go a little further. And so we did another mouse study where we took TCR knockout mice this time. So we can they don't control the infection long term because if they don't have any TCR, if they don't have any T cells, but it's a good way to show to sort of phenotype, because we know that the T cells that we're putting in are the ones that are functional. We can really look to see whether there's a change in their surface marker expression, which sort of infers like functionality. And so we, similar to the rag knockout, gave them about I think it was 2 million. Was it 2 million? Maybe it was 2 million adaptive transfer, 2 million T cells. And then we tracked infection 30 days and 60 days post transplantation and post infection with TB. And in the lung and spleen, what we saw is like some populations, there was a decrease in exhaustion. So CD4, TIM3 expression on CD4 T cells, suggesting that the mice are putting up a better fight. The mice that received the old plus mite are putting up a better fight. We then did a follow-up experiment with elderly CD4 T cells. So the average age from these were just commercially procured. I think the average age was 60, 65. We just chronically stimulated them with CD3, CD28 cross-linking antibodies because this was in vitro, to sort of stimulate or to show that this would have an impact also in beyond just the mouse. And so we stimulated them continuously for 7 days, and this is the response that we saw at day 7. So we saw improved activation, reduced markers of exhaustion in the elderly T cells. Again, all we did was we just gave them some mitochondria. 5 minutes in a centrifuge. So this whole time I'm doing these things, I'm like, man, this is so simple. Like this is some of the simplest science I've had to do it. So, but it's again the impact that it's had. We also took a look at senescence. Again, looked at the proteome that we had, and we saw that there were some shifts. And so we then went to again some elderly CD4 T cells and did just the typical stimulation that you would do for activating them, and then just followed them for 7 days. And again here, we looked at CD69 expression so as a proxy of activation that went up. CD57, which is associated with senescence, went down, and then beta-gal P16 double positive T cells significantly decreased. So at this point, I was like, man, maybe there's something here that we can use, because this is just for T cells. But again, because the mitochondria, they're different quantities per cell type, but they truly do the same things to different magnitudes. So if you could really scale this to try on different diseases or different conditions, I think it would be amazing. And this is just a quick recap. Really just saying that the mitochondrial transplantation reprograms AHT cells to function better. And I talked a little bit about the improving immunity. It reduced exhaustion and senescence. I showed a little bit of the influenza and tuberculosis data. And now here at Stanford, we've been looking at just different other conditions. So can we use it for glioblastoma as a sort of a dual strategy targeting both the immune cells, but also Trojan horsing the cancer cells with pathogenic mitochondria. And so when I got to Stanford, and then
Cole, hang on. I was trying to wait for the right time to interject, because the story is so great that I didn't want to jump in too soon. And now it looks like we've got a second chapter going, so I think it's a good time for me to. All right, so just to add a little bit more context, because I think when people hear CD4 T cells, their eyes gloss over. They really don't know. I don't know, average people, right? Or people who are unfamiliar with the immune system, right? So if you take lymphocytes, for people who don't know, if you do a blood draw, you'll get white blood cells. And then white blood cells can be further subdivided into neutrophils, monocytes, lymphocytes, eosinophils, basophils. Of the lymphocytes, now you can subdivide them into CD4, CD8, NK cells, B cells. So just to add a little bit more context, the CD4 T cells Are you familiar with Alessio Lana's work, the preprint on telomere rivers?
Man, look, that I am really interested in that paper.
Yeah,
Like it's very interesting. I
yeah, yeah.
don't I think I saw the preprint, but I haven't seen whether it's been officially sort of, given the final peer review.
It
But
may take, yep.
yeah, I'm inclined to believe that there's some like stuff there that is true. So
So he said that it's taken an average of 4 years for his papers to be accepted. He gets tremendous pushback. Just people just don't buy it. It seems too good to be true. But for people who don't know, in his study, in that preprint, which I'm sure will be accepted at some point, they rejuvenated CD4 and CD4 T cells. And as a big part of that story, mitochondrial beta oxidation, fatty acid oxidation, ATP production was central to that story. So for people who don't know, who haven't seen the video, basically rejuvenating CD4 T cells led to dramatic lifespan extension, like greater than every lifespan extension in any animal study, calorie restriction, rapamycin, you name it. So when considering the emphasis on mitochondrial function as being a key part of that story, your data ties beautifully into that, where instead of them giving the antigenic stimulus to donate, so the APCs donate the telomeres to CD4, and then the CD4 donate the telomeres everywhere else. And that gives you systemic rejuvenation. You're coming at it from, let's give the old and exhausted and maybe even senescent CD4 T cells, let's give them mitochondria, and that restores their functionality. So then the big question, even though the infection and boosting the immune system is amazing, because if you look at, and I know I'm going on a long,
no, this is a good podcast.
cool. So
We
so
plotted today.
I appreciate that. Cool. So when you look at the leading cause of death, or one of the leading cause of death in centenarians, top three, sure, cardiovascular cancer, but pneumonia. So the immune system and immunosenescence is a big part of you know uh, mortality at extreme older ages. So if you can rejuvenate CD4 and other immune cell types, whether it's through mitochondrial transfer or whatever, I mean, this is potentially a huge ROI on health span and longevity. So
That's
yeah.
exactly it. I mean, that's why I think this is why I get excited doing this kind of shit, because it's again, it's very simple, reproducible, and the impact, magnanimous impact. And even when I was in grad school, I remember because I did this. I was reading about senescence and stuff, and there was a clear link towards like mitochondrial dysfunction contributing to that, like to that signaling cascade. And I started thinking about, telomere therapy and telomerase. And I remember I was having a conversation with one of the other grad students, and I told him, I was like, man, look, someone's gonna eventually make the connection between mitochondria and telomerase. I'm not gonna waste my time. Someone's gonna do it, but so it seems like it's coming along. It's coming along towards like that trajectory. And yeah, to me it makes sense because we've seen it now with different senescence, both in from cancer, because we're doing some studies with that, and then also from infections. Like we see this improvement in the in immune cell function. So I think it's a no-brainer to me. To me, like I'm sold. I drank the Kool-Aid.
Yeah.
So
So wait, so not just I mean, basically all of the immune cell types, their function declines during aging, and I don't know if mitochondria function is a part of NK cells being less able to, engulf pathogens and B cells to produce antibodies. So
no, dude, it's all yeah. Like again, even without having I tell people like, okay, I have a background, I have a PhD in immunology, so I understand the networks. Understanding how mitochondria work is like a passport to understanding all the other cell types, because it's the same circuit in just different magnitudes. And so NK cells have a different function than a T cell per se, but the sort of the signaling circuit is really the same. And so you can make I don't have to study all these other cells to have like a sort of a predictive inference of how transplantation would benefit them. And I think what's interesting is also because um this is, and I think I'll have some slides on it at the end, but there's this, I'm interested in also seeing how to decouple oxphos from the signaling modality, because I think that's a beautiful opportunity to really control um like cell fate. Like it's clear that metabolites are super integral to that. And if you can control how much mitochondria you give, and the sort of like the the circuit that's within a mitochondria. That's going to completely change. Anyway, get excited myself talking about stuff, but this, yeah, exactly it. And because of the simplicity of this of transplantation, at least ex vivo, one of the things that I've been working at Stanford is how do I expand this to since I started here, for my postdoc phase, I did a cardiovascular fellowship. So I went from the immune system to trying to understand really how immune dysfunction drives pathology in cardiovascular aging. And so looking at macrophages, endothelial cells, vascular small cells, cardiac cells, all of them in the context of transplantation. What became
Wait, Colin, hang on, wait. I don't want to get too far. I got some more questions before you jump. Sorry. So,
you good, you good.
all right, just on the details. So, is it optimal to get so I guess the fibroblasts you got them from skin cells or
So the mouse studies, the fibroblasts came from MEF, like primary mouse embryonic fibroblasts. But then for the human studies, they came from at ATCC, like dermal fibroblast cell line, primary cells.
so skin. So if this is translated, and I know you're a part of, or you consult for Mitrix or Mitrix. I don't know. I
Yeah.
think they're doing some of this work. So is it the same setup where it's taking mitochondria from fibroblasts, or you're isolating fibroblasts from what? From skin, or where would you get the mitochondria from to do this in people?
So are you talking about in terms of Matrix or in terms of like how I would do, like if I were to move
How,
this?
yeah.
So I don't think it matters where the mitochondria come from, because if you can isolate them or if you can grow them in culture, and if you can, mitochondria respond to metabolic cues. So that means that in a cell culture, you potentially with the with CRISPR and the things that we have right now, you can program any cell to have the quality of mitochondria that you wanted to, as far as like bioenergetics or TCA like signaling circuits. I much more interested, or I think is going to be a point of concern, is the quality of mitochondrial DNA that you get, like from the cell source. And I would say getting them from younger cells, just pragmatically makes more sense, because it kind of anticipated that you're not going to have as many mutations and whatnot. But again, because we can culture cells ex vivo, like I don't see, at least right now, I have not seen anything to say that taking a mitochondria from a fibroblast versus a T cell versus skeletal muscle would dramatically influence, would necessarily change the outcome, if you can have like a sort of standardization after isolation. Does that make sense?
Yeah. So how do you foresee this then? So someone comes into your clinic or Mitrix. Well, I mean, granted, maybe they've got a different technique the way based on it. Sounds like you would do it differently from them. But so what? They're just gonna take a skin biopsy and then isolate mitochondria from the fibroblasts and skin, and then so wait, along those lines, what if and considering that, as you mentioned, mitochondrial heteroplasmy, right? So would you need to do function? I mean, granted, as you said, you can just bolus inject the whole mitochondria, but the biggest bang for the buck, as you alluded to with CRISPR and everything else, would be pre-selecting your best mitochondria previous. So would you do
The same stuff that we do for bacteria. Like, this is just bacterial engineering, but instead of the bacteria being in solution, it's like inside a glove box. So we have to just manipulate it that way. That's my perspective on it. I
so
think that it's Go ahead.
I was going to say, how do you so how would you pre-select mitochondria ahead of time? Because once you start hitting, once you isolate them and you start, I mean, can you generally after you give like complex one and complex two, I'm going, man, it's bringing me back to back in the days, but can you isolate mitochondria and then put them through functional assays, identify the best ones, and then transplant them in? But they're already basically used. You've used them up. You've consumed, uh, you know, succinate or complex one, things to donate electrons to complex one or complex two. Can you pre-select the mitochondria ahead of time while yeah.
Yeah, I mean, I think I would more so. We have a lot of omics information just scattered everywhere. I think that's a great way to indirectly infer the quality of mitochondria that you have per cell type, and then just kind of do your real functional assessment using seahorse or like flow cytometry. I think I don't see that as being necessarily a major challenge. Again, because um, if we have the ability to change how mitochondria respire based off of the metabolites that we put within a cell culture, then I don't see it as maybe like UCP1 because that's like a super like specific thing that only appears in a specific. But even with CRISPR, you can easily insert that stuff into non-natural places. So I don't see it as a major crux. So far I've been using fibroblasts because they're cheap. This stuff is, I mean, you see the state of science. We broke. Everybody's broke. So I designed my experiments again, for simplicity and also because trying to save a couple bucks. And so fibroblasts are the cheapest, DMEM, they grow, it's pretty obvious when they start to get senescent. So you stop that culture. So moving forward, I think getting someone's, some of their blood, maybe turning it into iPSCs, expanding them, and then sort of like selecting for the best quality of iPSCs based off their biogenetics, and then using that as the seed crystals for future transplantation is not implausible. To me, that seems like a pretty. I would try that.
All right, so I have some thoughts along those lines. So in studies that we've done on centenarians, are you familiar with dicarboxylic fatty acids?
Slightly, yes.
Yeah. So, so for people who don't know, you've got fatty acids that have a long carbon tail and then COOH at one end. A dicarboxylic fatty acid has a COOH at the other end. All right. So in studies on centenarians, we're seeing that levels of DCFAs relative to their parent fatty acid increase during aging, all the way up to centenarian status. So when mitochondria, and it's specific to certain carbon chain lengths for fatty acids, like medium chain fatty acids that become dicarboxylic fatty acids. So in terms of looking at biomarkers, like if you've got fibroblasts and you're looking, so you're just taking an aliquot from intracellular aliquot in order to assess functional status, potentially mitochondria. So the long story short is when there's mitochondria dysfunction, the ER and microsomes and even peroxisomes, the fatty acids undergo omega oxidation, which can then target them for excretion rather than having these fatty acids, which are going to be cytotoxic in high amounts because your mitochondria can't oxidize them. So I wonder if looking at DCFAs, intracellular DCFAs might be a way to pre-select for fibroblasts that have a better mitochondrial function. But you'd have to do, you said you have omic data all around. If you have, if you have metabolomic data on fibroblasts, that might be one way to pre-select without having to go through the, seahorse and, really, burning the mitochondria before you would put them into another cell or
Right.
before.
I mean, it would be like sort of like a satellite culture of you infer the quality from that. I think that's one thing. I for sure, and then moving forward because I see this moving towards clinical trial, within the coming years, and thinking about sort of the FDA, QC stuff that needs to be done. Uh, I think figuring out the best quality of mitochondria will be a combination of like omics information, but then also like functional testing. Not only looking at because I mentioned oxidative stress and ROS production is coupled to oxygen consumption. So really looking at the antioxidant parameters as well, and really the the portfolio of uh of mitochondrial ATP that is coming from pyruvate versus like amino acids or versus from beta fatty acid oxidation, having some sort of like you know metric or number or something that combines that information, I think will give a pretty robust range of healthy mitochondria.
One more question before going on. So
This is great. This is great.
yeah. So do when the two actually I have two questions, man. Sorry. So when when CD4 T cells incorporate the new mitochondria, do they discard some of the old ones? I mean, because if you pack a cell too tight with mitochondria, it may not be good, right? So do they get rid of some of the old mitochondria?
Yeah, so we didn't have the capacity to answer whether because we weren't able to track the mitochondrial DNA heteroplasmy after transplantation. So we don't have that information. Um, I think that some of it is staying, and that's based off studies that I've done now, based off just the amount of mitochondria you can deliver to a cell. It's impact stability, the retention of the liver DNA, and it'll make more sense maybe when I show this sort of like stuff in the future. But I think also, let me see if in this, uh, yeah, like right there. So there's also instances like this where we see literally on confocal, we can see the cells are trading mitochondria. Like so, I think it's a combination. I think some get deleted, some get traded because I think of a cell as a very intelligent thing because I mean we use them for thinking, right? And I think the cell has this ability to this sort of QC to say, oh, these are good, but I don't want to delete these. Hey, let me package it and send it across the street or something like that. I'm not entirely sure what all the mechanisms, but transplantation is not just happening through one modality. I think it's like it's a pretty like extensive network and loop. And so
All right.
some get deleted, some are probably getting shipped out, and then uh I'm sure that there's like secondary transplantation, which also is a benefit for the immune system, right? Because if you think about the cells that are constantly, kind of driving around the body, it's like it's immune cells. So if you can target the macrophages or these other cells that are like, patrolling, man, then they can do their secondary transplantation. That to me is a more pragmatic rejuvenation strategy than some, folks think that you have to deliver mitochondria to every cell in the body to have like and I'm like, dude, that is first of all ridiculous. Like I mean, you can try that. I don't think it's necessary because again, because if we can bioengineer or customize the engine before putting in the car, we can decide how much antioxidants, how many mitochondrial DNA copy numbers, like TFAM, these are things that you can send in the care package. So I think that's a beauty of knowing that we can like have like high fidelity transplantation. Um,
Wait, so then the mitochondria that are, I guess, donating some additional mitochondria after getting the transplant, are they doing that in vesicles? How do CD4 T cells communicate with other CD4 T cells? Is it exosomes or they're just budding off some vesicles to do that?
I think it's a combination. It's probably budding off exosomes, like mitochondria and macrovesicles. It's probably true, true nanointelligence as well, and also true the non-coded like extrusion into the milieu. We didn't particularly track the secondary methods, but from the literature, I mean, I wrote a book chapter sort of characterizing T cell and sort of immune system mitotransfer, and it seems like all immune cells just they just be talking like that. He's like, here, so I think it's a combination of that's allowing for transplantation.
Nice. And then how long does the effect last? So after CD4 got mitochondria, I think you showed up to 90 days with the infection model or maybe 60 days, but
60 days, yeah, 60, and then we had to stop just because these experiments were expensive. Like I'm trying to round out the paper. So I mean, I wish we could have gone longer, but like 60 days and we saw durability. And then for the immune cells that we did ex vivo, we looked at the exhaustion at 7 days and also 14, and we saw the same thing, like better, better. So that's like chronically stimulated cells for 14 days. But also there's this whole selection because of like exhaustion. So in vitro models for that are not the best, but durability looks to be like pretty strong. Again, we only put 2 million T cells. Imagine if you actually put a much higher number. So yeah.
how long do you anticipate? I mean, there's got to be a timeline beyond, and I get, you can't run studies forever, but where you'd have to do another transplant, right? I mean, are there plans to figure out, how long do you have to go before getting another transplant in order to keep CD4 or other cell type function higher than it would be at a given chronological age?
Yes, and maybe the studies I mentioned at towards the end will kind of give like a sneak peek next episode of Dragon Ball Z or something.
Huh. Cool. No
So
more questions for me for now.
no, this is good. This is good. This is like we're speaking electrons. So
Nice.
like I said, like I got to Stanford and some of these studies were done as I was transitioning from grad school to my postdoc. Really started thinking about if it is this simple, how can I be like Oprah and give mitochondria to every cell? Like you get a mitochondria, with enough specificity, but also because some cells in the body you really can't, you can't take them out. One thing that was pretty clear was that the other methods that were published on mitochondrial transplantation were kind of very low fidelity. This is fancy for 10%. Like, I don't know why they wrote it like this, but I was like, yo, 10 to the one is just 10. And these studies got up to 40 and 60, up to 40%. At least this is when at the time. And then even though our technique, this not really my technique, just the centrifugation method is good at increasing um like efficiency, you can't use this for cells that you really can't take out of the body. Like, and so for transitioning from just looking at the immune system and thinking about cardiovascular failure, the endothelium dysfunction or dysfunction of the endothelium is like a commonality among most of cardiovascular pathologies. And I was thinking about a way to essentially, use transplantation um, to help with that. You could do an infusion. You can just do, a systemic bolus of mitochondria that kind of probably look more like a bus where they would get to the destination, but maybe not to the right spot. And so we've been looking at a way to kind of make like an Uber system where we can actually tell the mitochondria with some fidelity, with some instruction where to go. And so I got some inspiration from, well, honestly, tuberculosis. Tuberculosis, it hijacks the receptor CD206 on alveolar macrophages to internalize. And I remember thinking, I was like, man, like mitochondria used to be a bacteria. I mean, they're kind of still are like quasi. They're kind of like cats. They domesticated themselves into our body, right? Like, it's the weirdest thing. Really think about it. But I was like, so what if we can make them, just more permissible to moving around? And so, um, engineering or like de-evolving mitochondria back into like its, ancestor just seemed a little too complex to me. And so I was like, let me see if there's a simpler way to just make them more internalized. And so antibody drug conjugates has been, they've been using this for a hot minute. And in most of the cases, it's to deliver a toxic payload to cells. So in cancer, based off the receptor, it gets internalized, kablooey, right? Apoptosis. And so we said, well, why don't we just see if we can do that? Like, can we just put some antibodies on the outside of the mitochondria? Maybe, just maybe it will act like a bacteria, but instead of getting, recognized as foreign, because they literally get internalized without, like I said, they're secret, these, we don't know all of the, without sort of uh, specific receptors, we don't understand all of the things necessary to have, mass internalization. But with this, because we can choose the receptors, then I mean, then potentially we can just, direct them this way. So what we came up with is a simple protocol where we have your donor cell type, whatever it is, you sprinkle some secret seasoning, you isolate the mitochondria, and you conjugate them with antibodies. And so this is the workflow. So again, my original workflow, was to get cells, isolate, and then spin in. And so now it's just to get cells, sprinkle, and then isolate. And so again, very simple because I'm always thinking about uh, anyone that wants to try this stuff needs to be like, easy. Again, still, even if there's internalization, like understanding the cell, like the fate of the delivered mitochondria, is it going to get eliminated completely? Is it going to force the native mitochondrial pool to get degraded? Is it going to be sort of fusion and like long-term retention? And don't have all those answers yet, but I'll show you some of the of our data that's suggesting, that there's potential long-term integration. And so first, I'll show just a comparison of the unmodified mitochondria versus like the mitocab or the antibody conjugated system. And this is comparing the internalization into CD31 positive endothelial cells. So we attached the anti-CD31 to the outer membrane, and then we just looked at uptake. And so we can easily again scale to 100% efficiency just by sprinkling the amount of antibody mitochondrial conjugates that you need. And most techniques that do the co-incubation transplantation takes like 24 to 48 hours. Dude, this is like 30 minutes, like 30 minutes in culture, and you're good to go. The same amount of time that you take to stain your cells with for immunohistochemistry or flow cytometry. So trying to make it as rudimentary because I know that there's a lot of labs that have data that says mitochondrial dysfunction, and they're like, what am I gonna do with this? Like, so this is an opportunity to really interrogate and potentially develop, I would say, therapies that are probably less toxic and multifunctional. Anyway, and so we're able to show that there's better internalization. We're able to also show there's better specificity. So this here is comparing a co-culture of the CD31 positive endothelial cells, which are like the translucent ones, and then the green ones are vascular muscle cells that don't express CD31, and then blue's a nuclear stain. And so this is by confocal Z-stack, and we're able to see that the mitocabs are very specific to the cells with receptors that they target. So easy to scale, specificity, great. I mean, at least to me, like initial experiments. And then some other data I'll show is sort of towards the retention, right? So in normal cells, they have functional oxphos. Uridine is something that's necessary to have a DNA production versus cells that don't have any that are deficient in mitochondrial DNA. They don't produce uridine, so you have to supplement them. And so these row zero cells are kind of like a great model just to test out like stability, long-term retention. And I guess like the question that we asked is like, well, look, if we can if these cells need uridine to function, if these row zero cells, these mitochondrial DNA deficient cells need uridine supplementation to function. If we do a transplantation with the mitocabs and we remove the uridine, and if there's proliferation and there's an increase in oxphos by these row zero cells, depending on how long it's suggestive of integration. And so this is just some of that data where we took wild type endothelial cells, we took the empty DNA deficient endothelial cells in gray, and then these are the cells that were treated with the mitocabs or the antibody mitochondrial conjugates. So again, we can restore oxygen consumption from 0 to 100. That's crazy. I never noticed that, but that's how, like, again, very, very like it's not rocket science. You just sprinkle the mitochondria on your cultures, or and we can show that there's also a restoration in mitochondrial ATP production from the row zero cells, and significant increase in overall ATP. And then we also looked at some of the proliferation, and we see there's sustained proliferation. I'm only showing 6 days, but I mean, we've got cultures that have been going now for like 6 months off of just one treatment. So again, the other question you're asking about, how much about retention of mitochondrial DNA and mitochondria, I think it comes down to the number, the amount that we deliver, and how much redosing we would need to do. But in these experiments that I'm showing you, this is just one dose. Like, that's it. Dose some, go home for the weekend. Like, you can come back later. So
question. So
yeah.
the uridine, and that's another one that most people probably know nothing about. So I think it's good to highlight uridine. So declines during aging, and as you mentioned, it can restore mitochondrial function in mitochondria that have like the row zero cells, right? So they don't have mitochondrial DNA, but uridine can restore mitochondrial function in that situation.
Uridine bypasses, so uridine is made in mitochondria and it supplements DNA replication in the nuclear compartment. So you have to because if the cells don't have functional mitochondria, they're not going to make uridine, so they're not going to divide. And so you give them a uridine in culture to sort of bypass the need of mitochondrial activity to continue having proliferation. So once you remove that from the culture, the cells, they stop dividing. They'll I don't want to say they become like senescent, but they really, a weird sort of senescence, because empty DNA deficient cells are really not a natural cell type to begin with. But this is just a good first pass to show that if there's any system to show that there's integration, as a first pass, I think this is the cleanest.
Definitely. But couldn't you make the argument that, sure, row zero cells are the extreme case, but I mean, mitochondrial DNA is being oxidized and degraded throughout aging, right? So couldn't extreme longevity be almost similar to a condition where some fraction of your mitochondria are almost row zero like kind of mitochondria?
I think, yeah, definitely. let me see. My first car that I had was a Ford Taurus. Like engine in a Ford Taurus. Imagine driving a Ford Taurus in 2026. That's how some cells in the body are behaving there, and it is in. I think it's role-like in the sense of the amount of pathogenic heteroplasm that they have is so high that the cells really can't function on oxphos. They have to switch over to sort of like I consider aerobic glycolysis kind of like credit card debt for cells because it's like they turn that on and then it's like hit one off. Anyway, so yeah, that's my perspective of that.
So wait, so along those lines, I mean, it seems like uridine can restore mitochondria in this situation where they've got depleted mitochondrial
Well, no,
DNA.
no, so it doesn't restore. It only restores the ability of the cell to produce replicate DNA, but it's independent of like if the cell if you can't if the uridine is made inside the mitochondria. So if there's no mitochondrial DNA, there's no oxphos. That means that you can't make any uridine, right? So you can use uridine to help boost, or I guess like helping detoxification, but I don't think it's the best method because again, like you said, if you have a car that's already messed up and then you're just providing it with things to help copy stuff, you're just gonna be copying bad templates, right? So I would not just use uridine by itself, but also again maybe when you start your intervention. If you're like 65, I think maybe uridine might not be, but if you're 35, maybe you supplement it and I have different responses.
Well, when you're 35, maybe you see no response because your mitochondria haven't yet hit that threshold for,
But it's
peak.
right. But that's what I'm saying. Like you maintain a better health threshold if you start versus like you start later. Your DNA is gonna help with proliferation. But if your mitochondrial DNA is messed up, you're gonna be copying messed up copies, right? Because even if we unless I missed something, but even when we activate mitophagy, it's not like we selectively just degrade the messed up mitochondria. I think it's ultimately it's whatever gets depolarized at the time. So I think we need some, I'm always thinking of like what's the most efficient way to have the cell think less about what it needs to do and just do it. And I think transplantation just fixes that because not only you're delivering the proper machinery to make the uridine, you're delivering better copies of mitochondrial DNA, again, the antioxidant enzymes, better phospholipids, better stable cardiolipin, all that stuff. Ultimately, because you can control how much. I think it's a much better way, much more elegant way to improve a system.
Have you compared the row zero with mitochondrial transplantation and then with uridine? And is it synergistic where they're working together to boost mitochondrial function, or the transplant by itself is just the bomb, and then uridine, you really don't need it at all?
So let's. So for example, here these cells are in uridine, like they're cultured in uridine, right? And you still have no oxygen consumption versus the cells that get the transplantation. So that's the difference because uridine bypasses all need for mitochondrial activity, and this is why I mentioned earlier about decoupling oxphos from the signaling capacity of mitochondria. This would be kind of like one of those ways. If you can decouple oxphos from the ability of the mitochondria to keep producing uridine, and you can then deliver that to a cell with specificity, I mean, I think that's a powerful thing, right?
Well, yeah. All right, cool. All right. Sorry. No more questions for now.
No, no, you're good, dude. This is like I said, it's much better when it's a yap session.
Yeah.
Because I don't know, I don't. Anyway, so this is the kind of framework I've been thinking about with this technology or like with this, I really, I mean, it's just antibodies. Like I call it technology, but it's, because we can because we can get a much higher delivery efficiency, like 100%, and you can read those because you can choose different receptors. I think the sky's the limit as far as like the potentials. But just really manipulating the cellular genome and the mitochondrial genome to either insert things or remove things using CRISPR or other gene editing tools. Then isolate those mitochondria. You can even insert synthetic mitochondria outside of the cell. I think that's also a beautiful thing. Repackage them and then get them ready for delivery. I think is a very powerful way to control cell function because you don't have to just rely on one specific kind of mitochondria. If you can if you can really customize and develop different sort of enzymatic circuits, then maybe you can truly control the fate of a cell. And we talk about this in science all the time, right? Like we learn about these pathways in sort of like a linear thing, but it's really like that movie, like, everything all at once. It's really, and so I think to have a much more durable, like functional change, you need to have multiple pathways being innervated at the same time. And this provides that. If you can decouple oxygen consumption from the system. So again, it's really just, and how I think about it is reprogramming the bacteria. And because if you, for example, with these cells, like these rosier cells, where they have no mitochondrial DNA, they have a finite time. Like you can engineer them so that they're not gonna integrate. You can change the ligands on the outside so that they're more prone to mitophagy. So it's really, I think, a very like concerted way to have something happen in the cell and then be like, all right, skedaddle. Because that's really what that's medicine, right? That's what we ultimately try to do with drugs. This is a living drug. I really do think that this is um so maybe some people are gonna think I'm more crazy after this
Yeah.
after this presentation. But along the lines of then, realizing that I think this is a very efficient way of getting mitochondria into cells. It's kind of, if you have an Uber, then you got to have like a Google Maps, right? And so this is something we've been working on, figuring out what's the right receptor combination, or like um not only receptor combination, but like mitochondrial damage threshold necessary, or like important to consider to sort of have a picking order for which cells to target. And with this system, because it's again, because you'll have the ability to really it's not going to be, like 100% specificity, because receptors you're going to have some overlap. But I do think that because of the similar to other ADCs, we should be able to have a lot of targeting potential with the system. And so I think that this could be influential for almost anything that involves mitochondrial dysfunction, whether it's something that's like aging associated that pops up, or something with primary mitochondrial disease. I've been focusing on immune aging, but I mean, it's all connected anyway. So immune aging influences all these other things, and these other things also influence immune aging. So targeting mitochondria, whatever the cell is, I think is a powerful thing. So towards this, shout out to Burroughs, man, because they were the first ones to believe in in the craziness. But through this grant funding, I've been able to really kind of build a proper repository, or like picking order for which receptors are worth targeting on different cell types. Um, this is we're talking about the immune system, right? Like, so we've shown that we can take immune cells out the body, CD4 T cells out the body, give them mitochondria, and put them back inside, right? And I think that's great, and I think that's safe. But we don't. Why do we even need to take them out if we can just target them in vivo? So this is an example of that. We're working on making a immune specific. So CD11A is a receptor found in all immune cells. Here we're showing that they get into like the mitocabs get internalized by them, and this is the efficiency compared to unmodified mitochondria. And again, this is only after I think this is like 90 minutes to 2 hours. So significant 100% internalization, and less is more. You can use less to get a functional benefit. Another project that we have that is ongoing is looking at aortic aneurysm development, which is smoking and aging associated. And there's a lot of gene signatures suggesting mitochondrial dysfunction in both the cardiovascular and the immune compartment are like, it flares up. And so with AAA, as they call it, it's a giant game of who done it. Nobody really understands what's the first cell to sort of like pop off all the damage. And I was like, dude, mitochondrial dysfunctions in all of them. Let's just give them better mitochondria and see what happens to the system. Because from the dearth of literature that's out there, you give cells better mitochondria and they just kind of figure it out. I don't think this is gonna, again, for people that are listening, this is not gonna get you immortality, I don't think, but at least it's a way to stave off a lot of the aging associated, like, signaling, like signaling dysregulations that sort of contribute to cardiovascular disease, immune dysfunction, neuropathology, etc. And then this other project here that we got a pilot fund from NASA for looking at, if I want to go to space, I think it was Mars for a minute, and now maybe it's the moon. But anyway, like our physiology is just not, from the amount of literature that's coming out from NASA, our physiology is not supported for long-term space flight. And a major signal, a major pathological like signature from space flight is mitochondrial dysfunction in all the organs, pretty much. Like they surveyed a bunch of them. Mitochondria are wonky in all of them. And so we said, look, or I guess I said, look, I mean, this is not a way to, again, you can't stop the radiation damage because it's the match. Just like you can't, if you're in a spaceship and you're out there, you're gonna get. But if we have a way to provide astronauts with a sort of like a shielded, mitocube that they can then resuscitate and give themselves. I guess this is where people start saying that it sounds like sci-fi, but this is like a next week thing if we really keep moving. Like they have a way to then just um, um, boost their mitochondrial health. And again, because we have the ability to really bioengineer or tinker and tanker and build better mitochondria for those environments, this is a real opportunity for improving like our physiology beyond natural capabilities. So yeah, this is just some pretty cool stuff, I guess. And then you're asking about sort of dosing. And so this is our project that we just wrapped up, where we had 6 age marmosets and two six it was a total of 8 marmosets. Two got just saline for 8 weeks, and then 6 of them got a weekly dose of the CD11A. So this conjugate for 8 weeks, once a week, and we've collected um, serum and we've collected immune cells every 2 weeks, and we've done a PET CT before and after the study to really see what kind of durability this transplantation is having. So maybe this answers like what I've been thinking about towards like the future and moving it forward. But I mean, I think we're at the doorstep of something, something pretty cool.
All right, wait, just to chime in on the marmoset. So uh, it wasn't a survival study. I mean, you're just looking at the impact of the mitochondrial transfer on just basically cellular function, or what was the endpoint? What were the endpoints that you're going to look at?
So the bargain, or I guess like the the way I was able to get this pilot funded is that I kind of used like the logic from stuff that I did in grad school / early postdoc, where we showed that we could take immune cells out and give them mitochondria and put them back inside the body, and the cells work better. And so the next obvious step would be to uh, give mitochondria directly to the cells inside the body and see what happens longitudinally. For this study, the stipulation was that we could not euthanize the animals. So we could just really look at kinetics. And so again, simply designed to understand from a perspective of, especially if this is going to be something for like the aging populace. The marmoset age group was 10 years or older, which is comparable to like 60, 65-year-old individuals. So it will give us a pretty good understanding of dosing, I would say. How much mitocaps can we give per week? How much inflammatory response are we anticipating, if any? Like if there's a systemic change in the frequency of circulating immune cells, like if there's less classical macrophages circulating, less interferon gamma producing T cells or CD8s that are just like wonky, being cytotoxic for no reason, just cause like they're flustered. I think that's a beautiful thing. And so I'm waiting to get some of the samples or some of the data where we're going to test. We're going to try to phenotype the immune cells, look at the frequencies across time to see what we see. But this is just the first step in that exploration.
And then is the plan, so I know I broached the subject earlier, but staying in academia versus doing this in a corporate space? I mean, because I think you got to stay in academia and really fight this fight for, until you've mapped it out and, so that it becomes a real therapy that can be applied clinically, especially in aging or even age-related disease or any disease. So what are your thoughts on that?
I mean, man, it's a jungle. First of all, I've benefited from the academic environment for sure. Like, I don't think I've been able to do kind of science other places, and um, at the same time, getting funding for this, like it's been ridiculous. I can't. I've got more grants calling me stupid, like literally, like this is some bullshit. Than even entertaining the idea. So for the last 4 or 5 years, has been ridiculous, and so The reason I've been able to get these pilots funded is because of, you got to be able to sell the science. And I'm hoping that within, because I'm not the only person doing this kind of research. I'm hoping over the next like couple, maybe year or two, that there's more papers that come out that provide support towards not only transplantation, but sort of like in vivo uses of it and systemic like applications. Um, with that said, like, I mean, it's not cheap. Single cell sequencing, it costs like 2 Teslas just to do a couple samples, right? Like it's insane. So it really depends on where I can sort of bootstrap to get, funding from. Like if I can write more grants and get steady funding, great. But also I have to consider, like, if it's easier, if it's the path of least, electron leakage per se, if it means like gotta start a company and go that route, I'm not opposed to it, because I really want to see this get to the clinic. So that's the main goal.
What are the crowdfunding rules at Stanford? Is that an option at Stanford where, you could
I
have
think,
people?
it's definitely an option. I mean, I how I think about it just from the uh so I know for sure that there's like an office of philanthropic like donations at Stanford and stuff like that, and it's easy enough to, if that was the case, like, and there was a crowdfunding like push, I would contact them, put the folks in contact, and they'd be able to set up a, it's like a research agreement or whatever. And I think there's also, you can also um provide funding in terms of like a donation. There's some stipulations, but it's definitely allowed. Especially now with the changing landscape and people realizing that you can't rely on federal dollars for funding. That people are really having universities are really having to rethink how they approach um, funding. And it's going to be towards IP. Like, that's, to me, the clearest thing that's going to happen is like a lot of universities are going to start spending a lot more time sifting through IP to make sure that they have something that they can pull interest from, either from philanthropic, but also from industry collaboration. So yeah.
If individuals wanted to like donate to your work, so they'd have to contact, do you have like a link that I can include with the video so that if people wanted to donate to your work or donate to Stanford, who would give you a cut of that? Is that something that could be possible?
Yeah, I'll email you the link. Let me see if I can find it right now.
Yeah. All right. So risks, what about risks? I don't think and that's basically, that's the last thing I got is what are the
Hmm.
risks of mitochondrial transplantation? Is there a balance between you inject too much mitochondria or you too many targeted mitochondria, maybe you increase cancer risk, but then people will say, oh, cancer is fueled by, anaerobic glycolysis. It's not a mitochondria story. So maybe that's a low risk proposition. But what are some risks for the mitochondrial transplantation story?
So right now, but based off the literature, and I can tell you from the experiments I have done, all have been allogeneic in nature, meaning for the marmoset study, they're allogeneic. We haven't seen an inflammatory response so far from the data. The experiments with primary mitochondrial disease fibroblasts that we've done are also allogeneic, and we see long-term like pretty much engraftment, like no net negative so far. But we haven't done extensive enough sort of omics to look at all of the changes in the transcriptomic profile. I think there's hot debate about whether a mitochondria, say from me to you, would last, like if it would have like a long-term impact, or would it just like be there for a couple of days and then Scooby-Doo. I think it comes down to how much you can like deliver and establish sort of like a seed crystal, right? Like if you have established a network, I think the mitochondria, they're doing some sort of communication. I can't explain it with enough data, but like they're talking, they're definitely talking to themselves. And so the major risk that we tend to think about is, so healthy non-dysfunctional mitochondria do not seem to induce any sort of pro-inflammatory impact. There's a study that looked at they induced sepsis in mice, and then they just did a tail vein infusion of mitochondria, and sepsis, like systemic inflammation, completely disappeared, which is insane. But I mean, the data is there, suggesting that it's the quality of the mitochondria as far as like the how stable it is. And so we're thinking about um uh uh like QC processes. Like, how do we make sure that that the isolation process is not in having a lot of damage happen or exposing things from the matrix into the outside. So those are things I think about. Heteroplasmy has not been a major uh I guess like contention point in my head just yet because so far I haven't seen any data to suggest against it. But I know there's Monovia, there's other companies that are doing transplantation and they might have different views on it. What I think about towards I was asked once, does it need to be autologous or allogenic? And I said, I mean, we can make both. It's really not expensive because if you have a cell that's printing your mitochondria, like you can standardize that. You can take someone's blood and then literally like these rosary cells, these empty DNA deficient cells, they kind of in most situations they take up and start proliferating and integrate whatever mitochondria you give them. So in that system, you have a printer. That's a printer right there. Just the major difference would be just the sequence of the mitochondrial DNA. And I think you can sort of use like familial tracing, maybe take haploid groups that are similar like to your own lineage, and you might not see as much uh, um, immunogenic impact. But again, I'm not a physician, so can't answer just yet. But I don't see that being the major issue. I think it's more so going to be just the preparation quality. How healthy are the mitochondria like at infusion? That's what I've been thinking about.
Why would people get mitochondria from someone else? I mean, why not just get it from themselves? Like you said, allogeneic, and
Well, so okay, so if you're, for example, if you, it depends on, it intrinsically depends on the level, like your, if I've been smoking crack for 20 years, right? Extreme example, smoking crack for 20 years, and I work in a coal mine. At 60, I think I'd have a better chance finding familiar mitochondria that is a better quality because the amount of sort of enrichment you need to do, and this is all hypothetical, maybe it won't be that much, but the amount of enrichment you need to do for your cell population just to find that subset of cells that has healthy mitochondria, and maybe you can use T cells as a way to sort of select for better quality. But then a T cell is so the amount of mitochondria in a T cell, so you need to then think about de-differentiating them into a different cell that has a much higher capacity, right? Um, to me, it just seems a lot simpler if you can just get and that's it in that situation. But if you're young and you're healthy and you want to bank some mitochondria, if bank some immune cells, really, there's nothing wrong with that. So that's what I think is it would be sort of like a case per case per situation. One of the projects that we're working on right now, which is really going to be a sort of autologous situation potentially. Um, uh, it's looking at a prime mitochondrial disease, and there's a mutation in FXBL4. It induces like hypermitophagy. And so, one of the strategies that we're going to do is we're just going to correct the hypermitophagy in the patient's own mitochondria, and then just redeliver those to see if there's like longer retention and better like functionality. So in those cases, like you could use your own, but um, yeah, it's a case by case basis. But another thing to think about for people talk about this the cancer transfer, right? Like I think it's again I understand cancer, but I also understand oxidative stress, and I think it's more so that cells have some sensing mechanism for ROS thresholds, and they donate mitochondria because of that. I think that's pretty much what it is, and so there was also a study looking at cancer and showing that the heteroplasmy in cancer cells is completely different than the heteroplasmy in immune cells after they do the whole telling on the TNT system. So, do mitochondria really belong to us? Like, are we really like our own mitochondria really ours? Really, like I don't. It's a philosophical question too, but I haven't spent too much time beating my head about it.
nice. And then what about man, I had another question in there. Where did it go? Doping. So right,
Yo, but yes, dude, doping. That's something I think about a lot because I know I can tell you right now, as black market, yeah, people are gonna be blood doping. Oh, they're gonna be mito doping because it's gonna be so hard to detect. I mean, this is something I've literally been thinking about from the moment when we started doing transplantation because we've done studies with looking at peripheral artery disease and sarcopenia, and we saw significant recovery after we injected them. I was like, oh man, people are gonna like, it's gonna change a lot of things, and I don't know if it's gonna be necessarily a good or bad thing. If you want to run a marathon, maybe you can, train faster, help with recovery in that sense. But I think it's definitely gonna challenge some of the ethical um standards that we have right now towards athletics and just like life. I mean, I think I see this being something like, you don't need a prescription to get mitochondria. You can go get some the same way you can get some ibuprofen because there's such our body literally breaks stuff down to make mitochondria. Like it's just to me, it's just some inefficiencies that we could maybe make better. Yeah, blood doping is something that really, really interesting. So
It's going to be unstoppable, like you said. How would you detect that someone got a bolus I mean, you'd have to do like a biopsy and pick your muscle of interest and then look at that, do another biopsy. No athlete's going to want to give a biopsy in the first place, but
and it's going to be very hard to differentiate between, at least I don't think there's going to be a major difference in showing natural um hypertrophy from working out versus like injecting mitochondria because they work on the same sort of pathway. So I don't know if you'll be able to differentiate unless you were to take someone else's mitochondria, you have to sequence. But then depending on the amount, like you'd have to do a lot of sequencing. It's gonna be very tough. It's gonna be very, very tough. So
yeah. So in
yeah.
terms of rejuvenation technology, in my top three are mitochondria transfer, the stuff you're doing, Michael Levin's bioelectrical restoration, and then the telomere reverse story. So, and most people, I mentioned it, in the intro, that most people are only thinking about epigenetic reprogramming. But how do you see, like, sure, you're studying mitochondrial transplantation, so maybe you're biased in this, right? But as the objective scientist, when it comes to human rejuvenation and restoring youthfulness, not maybe not totally, but at an advanced age, would you put mitochondrial transplantation close to the top, better, even better than epigenetic reprogramming, or how do you see that?
I think the risk profile is definitely lower for transplantation than epigenetic reprogramming, just because of I think you have to have proper sort of pulsing and time constraints, so you don't induce too many oncogenes. And I was actually thinking about this too, like if you can customize a mitochondria, then maybe you can just use that as the transient sort of reprogramming vehicle. So the moment it gets to the cell, it like induces it, the enzyme circuit produces what it needs to, and because it's marked for degradation, it doesn't stay long. And so I think that's a much better way of controlling reprogram. Because again, I'm not a complete expert on reprogramming, but from what I understand, it's finding the right sort of range for exposure of the different reprogramming Yamanaka factors on cells. And I think because we can program stuff to have mitophagy happen, you load the mitochondria with these things, or not even load. You don't even need to load them. You can just co-conjugate them onto the outer membrane, and over time, the mitochondria are either going to fuse, but you can also again program them to not fuse and just be headed straight for mitophagy. So the the reprogramming happens, the mitochondria, get gobbled up after. And I think that's a durable way to do it. Right now, I think there's a lot of issues with specificity, like because of this whole, the same with like, with antioxidants, every cell has a specific Goldilocks kind of range for how much they need. And I think it's the same thing with reprogramming and trying to understand those gradients from flooding a system is just it's hard, right? So having specificity, mitochondria are literally like our personal bacteria. Like we should really be thinking about it from this perspective. Changing how we, and the fact that we have so many in us, dog, like it's so cheap. You can grow so many, you can really customize them, treat them like, re-engineer them, package them with different things. I think it also because our body has an intrinsic, they accept them. Like you don't have to do much. Like even without these modifications with antibodies, like if you were to incubate cells for 24, 48 hours, they would still take up the mitochondria, suggesting that there's already some original sensing mechanism. This is just trying to make it better.
Amazing. This was great. Thanks, Colin. I'm looking forward to, I mean, you showed a bunch of studies where, I think through 2029, you're funded through 2029. So I'm looking forward to seeing that data. And yeah, thanks for sharing. And I'm hopefully I can host you at another point or more points in the future.
For sure, we can have a follow-up. This was amazing. This was I appreciate you also taking it, because you have to push your schedule back a little bit. But no, I like doing this stuff.
Cool. Thanks, Colin. Have a great day.